Let's recap on a 3D comparison. Again, there's a way to look at proteins in 3 dimensions, in 1D, 2D, 3D. 3D is the 3D coordinates. 1D is just a string, sequence, secondary structure written in a string, solving accessibility. 2D, the interest, yes. Can you try it again? I heard exercise
session on Thursday, will it be?
Oh, the exercise session will not be on Thursday, no. Because Thursday really is a public holiday. So again, the 2D representation, the inter-residue distances, gives the same information except for the mirror image. Dynamic programming is a way by which you can compare sequences. It's brute force. You essentially try all possible combinations. On this matrix I've shown you, on this matrix, this is the straight path. You can also allow for gaps. Allowing for gaps in the matrix essentially means you turn right. Then you, at the end, have the decision to make whether you take short alignments, local alignments with the highest sequence identity. This was 2 and 100%, or larger ones. I had this number 4 and 7 there. Ultimately, which one is the advantage is decided by this function here. And the function typically uses an exchange matrix or a scoring matrix. I presented the Blossom matrix to you, Blossom 62, where for every single pair of residue you read off in this matrix how much you gain from aligning these 2 residues or these 2 amino acids. There are many of these substitution matrices around these days. That's certainly in a thousand. Most methods really use this Blossom matrix that is compiled on robots. Here is a method, a tool that you can play with that sort of allows you to play a little bit around with dynamic programming, developed by the lab of Francesco Mello. Now, I also said that this dynamic programming is slow when you do large-scale comparisons. We don't want to speed it up. We speed it up by fast hashing, finding single words that match, and then extending these matches. The major challenge in this is getting the statistics right. So essentially seeing where here in red on the right, the real score sits, just a minute, and how far the real score in red is away from the blue, the background distribution.
Could you please explain again how we come up with these additional words in the class algorithm?
So I'm not entirely sure what you mean. So let's begin with the idea. So the fast hashing idea is you store all triplets, so all words of 3 letters in the database. Okay? And then you have a rapid way to hash running through a sequence, is this word present or not? Now, if at 2 positions you find this word, you have a match. Okay? And this is what I indicate here with the initial seeds. So in this particular case here, in the initial red alignment, I have 5 seeds. I have 5 identical 3 mirror words. Again, in the real blast, it's not identical words, it's words according to some cutoff in the Blossom matrix. But these are identical words, idea. And then you extend the match. You extend the match by dynamic programming. Now, dynamic programming is still very fast here because you now do not apply it to the full length. It's essentially n squared, so the length of the protein squared or L squared. But now, since these words break down the word, just assume you find one hit only in the center or somewhere in the middle of the word of the entire protein. Half times half is a quarter, so you quartered already the CPU. But just a single word hit, you can still do dynamic programming afterwards, but you have a quarter of the time. But in this particular example, you already found 5 hits, so you will break it down by much more than just a factor of 5. And ultimately, it's not quite true, but ultimately that you break it down almost to the level of a linear search algorithm, linear in the length, a little bit more. Because the extension costs a little bit, but that depends a little bit on how these words are defined, how distance, there's a variety of reasons or conditions here that depends on. But it's clearly much faster than n squared, or L squared, right? Did that answer your question?
Yeah, So we're not using any seed words in particular, but just look at all the possible.
Yes, which is in the space 8,000. So it's 20 different amino acids. 20 by the 3 is 8,000. Is one number. It's easy to try. Yes.
And we're just performing one linear scan,
yes.
Not accounting for shifts.
Yes. Well, wait. One linear scan is accounting for shifts. You're looking wherever in this sequence is that word. And if you find it in the other one, you overline these two.
I mean, you're like aligning zeros and zeros, and then you're going like one sliding window, right?
Yes.
So if, for example, YKL is here and YKL is in the end of other word, these are not going to be matched, right?
These are going to be matched, yes. But now assume, so let's assume that there is a hit here, and the same hit, YKL, maybe it happens to be somewhere else. Say this would be YKL as well. It's not quite the end. But then this match would be written down too. And then which of these two, so this will be in what I report here. What I report then would only be the first of these two lines. And there would be a duplication and it would report another of these two lines. And then would see, can I extend the second hit? So if that would hit here to there, is there anything else in the vicinity? And in this particular case that I show, you immediately see there is something in the vicinity. So immediately this would sort of come up as the better hit. But in principle you do them all. But again, this is, you pre-store that in the database. It's very, very, very fast. Okay, that's the hashing part. Again, the major problem here really is to get the statistics right. Or to get the statistics in a way. So the word right here, this is a hack. So BLAST compiles the background statistics, estimates the background statistics. And it works very well. But if you would really simulate the background statistics for every single case, then that in itself would take a long time. Now, we go to the level of multiple sequence alignment, but before we go, let me get back into this question, how accurate are pairwise alignments? Dimitri, the camera has been running all the time, right? Thank you. So, rule of thumb, when you align two sequences, 100% sequence identity is trivial. You know that they are related, they have similar structure, they have similar function essentially. Okay, very simple. So then you go further down. So the similarity between these two sequences, and I'm going to talk about percentage sequence identity, so the percentage of amino acids that are identical between the two. So it's clear that at some point here it's no longer trivial that two proteins are similar in sequence, in structure and function. Still, there's a region that we typically refer to as the daylight zone, where the comparisons are such that if I have this level of sequence identity, these two proteins essentially are similar. Okay? And you will observe that the number that I put here, or the yellow, the daylight zone, extends very far. Two proteins that have 40% sequence identity essentially typically have similar structure and function. This is surprising actually. So they differ in many details, but coarse-grained the structure is similar. This is surprising because with 40% you change more than half of all the protein. Is that surprising to you? It is, right? What do you believe could be the reason for that? So actually, I don't believe, so we haven't really done the experiment, we have done it for particular examples, but I actually don't believe that if you completely freely changed, you took a protein in your lab and you changed every single amino acid, that you would create this, that you would get that far. I mean, yes, you will find solutions that get that far, but you will find many solutions where you change structure and function, and you change much less than 60% of the protein. So how come we observe that so much can be changed, and it doesn't matter, seemingly? Any idea? What could be the reason for that?
maybe the function is driven not by the entire sequence, but by some parts, essentially.
That is true. Function is driven by some parts, but this is correct. Unfortunately, we don't know where these parts are hidden. So when you take a stretched out chain and you fold it up, so you can, for instance, imagine that these binding sides here are important. So these two sides, but in terms of sequence, they're far apart. So where to find them may not be that easy. I'll get back to that briefly in passing, motifs. But what could be the reason why there is such a pressure to keep it? Yeah.
Some regularities and repeats are more important than the.
Oh, that's an interesting idea. So the idea here, for those of you who sit very far away from him, maybe there are regularities, and these regularities are repeated, and repeating regularities gives me a simple alphabet or a simple way to construct things, and that's why I see them again. In some sense, that's true. In some sense, this is why from some point next week, I have to redesign my lecture. I believe certainly next Thursday we will begin to talk about 1D structure prediction. And there these kinds of motifs will exactly be found. They exist, but that is not the explanation for this one here. There is a pressure underneath. Any idea what that pressure could be? This is sort of the result of a selection process. Now I use two words that sort of somehow could give it away if you knew the answer. Any idea? This is evolution. The point is that if the protein is not doing the right function, the probability of not being seen is very high. I'll put it differently. It will survive and be reused only if it does the right thing. Changes happen all the time. Radiation, the duplication mistake, there are many reasons for change. The changes that are neutral survive. The ones that don't. There are some changes that in fact improve things, but they're very rare. But that's the reason why you see the daylight zone extending that far. Because we are comparing objects that evolved under evolutionary pressure. And the evolutionary pressure was to maintain function. Okay? Yeah? So daylight zone, the idea again, what I'm asking is, I talked about dynamic programming, I talked about fast hashing methods to compare two sequences. Now the next question is, this is a 1D comparison of sequences. At which level of sequence similarity, identity, or alignment score can I say that they have a similar structure? That's the question, right? And I'm beginning to sort of look at it in terms of zones. So when two proteins are very, very, very similar, 100% identical, they have similar structures, that's clear. And at some point, we will possibly no longer be possible able to say that two proteins have the same structure. Daylight zone is defined as the zone where from sequence similarity, so all I compare is protein sequences, and from their similarity, I will be able, without mistakes essentially, to say that these two proteins have the similar structure. I may not know any of the two. All I need to know is their sequences. The sequences are similar enough to say they have a similar structure. That's the daylight zone. Okay? Yeah?
Doesn't this argument maintain function only hold when we look at homologous proteins, not proteins in general?
So that's the next question here. When we compare any two proteins from the database, we have no idea about the evolutionary relation. So the argument that I'm building up here, that evolution is pressuring, is the question, is that argument only valid if I look at homologous proteins, two proteins that evolved from a common ancestor? That is exactly right. You're 100% right. In the daylight zone, however, this is exactly the kind of the similarity. Although 50% of the amino acids change, typically they evolve from the common ancestor, these two. So typically, in this realm here where we are in the daylight zone, we would typically assume that they are homologous. Exactly. It may be surprising, but this is, you compare many sequences and you find that the ones you find at that level of identity, let's call it an empirical finding, the ones you do find, pairs you do find at that level in the daylight zone, they are evolutionarily related. Okay? Then we enter a zone coined twilight zone. Very simple, right? So you want to infer from sequence alone that two proteins have a similar structure. This signal sort of fades. It disappears, and then there's a midnight zone. The midnight zone is simply the zone where the sequence similarity does not tell you anything about structural similarity. Because essentially we are down to levels of random similarities, and you cannot distinguish between the signal and noise. The midnight zone is in fact the one where most proteins of similar structure sit, but that's a longer story. Okay, now I'm going to show you two views here that are not real data. It's just to point out an expectation. So mind you, this one here is 35%, so previously the daylight zone is somewhere here to the right. Going from 100% to 40 is where I sort of roughly put it. Now the question that I'm asking is, upon entering the twilight zone, how is the signal going to disappear? And what I'm showing here is true pairs. So true pairs are proteins that have similar structure. So the more distant sequence goes from 15% identity to 35% identity on this graph here, the more distant, so the more to the left you go, the more the signal will no longer be right. So you will not be able to infer from sequence identity that two proteins have similar structure. That's why you go from 100% in the daylight zone, 100% of the pairs that have more than 40% sequence identity essentially have a similar structure, to something like 50%. Again, this is fake. These data are not true. Just to illustrate. At the same time, we expect that in the same sort of transition here, we would now plot the number of pairs, again sequence identity down here from 15 to 35%. The blue is what I showed you before, pairs of proteins that have similar structure. Red are proteins that do not have similar structure. So we expect that in this twilight zone, the red is going to shoot up. And at some point, so red equaling blue here means that your accuracy is actually 50%. Half of what you find there is blue and half what you find is red, so 50% accuracy, right? How can we produce the data? How can we check how the real curve looks? Well, one way is, actually the only way is, we begin with proteins of known three-dimensional structure. And now we do a 3D superposition. So we take all the proteins for which we know the three-dimensional shape, and we superpose the shapes. And then we say, okay, some of these shapes, they are essentially the same, or they are similar in 3D. Some are really different, and some I'm not sure how to say, I ignore them. Somewhere in between, and the value that I put down here is the root mean square deviation between two proteins. So if the root mean square deviation is 2 angstrom or 0.2 nanometer, then I say they are the same. If it's more than 5 angstrom or 0.5 nanometer, then I say they're different. And in between, I ignore all the cases. Okay, now I have the red and the blue. I have a set of pairs of proteins that are similar in structure and different in structure. Okay, now all I need to do is do the same pairs here and align them just in sequence by the algorithms that I showed you before. Dynamic programming or the fast hashing. Now, the question is, can I do what I put down here? So can I take the entire database of all known structures and compare them all against all? This would be the optimal, in terms of size of the data set, the largest possible data set that I can get today. But actually, it's not the right way of doing it because databases are biased. So this may be completely biased, what I see there, by, so half of all the known structures, in fact, if I just took, I start with protein 1 in the PDB of known structure, and I say that I add 2 if and only if it has less than 100% sequence identity. And by this algorithm, half of the PDB is thrown out. Because half of the PDB are proteins that have 100% sequence identity to something that is already there. Could anybody imagine a reason why that would happen? In fact, there are many reasons for that. Some are social and then some are scientific. No idea? Well, yeah?
Maybe two groups have added the same thing.
That's a very, very important reason. And it happens all the time. Let me just repeat to the people in the back. Before I repeat the answer, let me just point out again, today a protein structure, to put one record to the PDB, the protein database of known structures, has about 100,000 records. To put one more costs about $100,000. So imagine there are two groups going at the same time for a hot structure. And in fact, there are more than two groups, and they all go for the hot ones, because the hot ones are published in the best journals. One is going to be the fastest. And the other one is also going to put it down, because after all, there's $100,000. They publish a journal lower, but at least they put it into PDB. That's one important reason. So competing groups. Another reason. Yeah?
Maybe there is already some hidden structure in proteins that enables us to determine the PDB structure.
That is sort of a similar one in some sense. That he said there is a theme in there. So let's just shorten it and twist a little bit what you said. You could do it again, it's easier if you do it again. So it's not the competition idea, but there's something in there that makes it easier to do that structure again. And that is the second most important reason, or maybe even the first most important reason. These things are difficult to take apart and to count. There's another reason where people just do a better structure. So there was one in the past that is not as highly resolved. Now there's a much more detailed structure, and those you find very often. Then there are repetitions where people do folding studies. How fast is this going to fold? And every single time they get a new structure, they put it down. There are studies of the protein binding to something and not binding to something. The sequence is the same. It's just the binding partner. But the protein is not changing, right? And again, this is some very, very important scientific information. How does binding change the shape of the structure? How is the molecule that it binds bound? So these are all reasons. Anyway, so we have to reduce the redundancy. And to reduce the redundancy, this is what I sort of indicate here. I take this white part. The white part is such that no pair in this white has sequence identity above a certain threshold. That's one definition of redundancy reduction, right? Now, what I do is I do an alignment of all against all. So I take a redundancy pair out, a set. So this is 100,000, the redundancy maybe 10,000. I take this redundancy set and do an all against all of the 10,000 against 10,000. And then I'm going to see how it happens to enter the twilight zone. Is that possible? It's not. Why not? There's a mistake in what I just proposed to you. I lied to you, so to speak. Yes, I have to absolutely reduce the redundancy, but I made a mistake here. Can anybody spot the mistake? The redundancy, reduced island here, or whatever you call this little thing, the subset, is defined that no pair in there has a sequence identity above a certain threshold. Let's just make it more dramatic. No pair in there has more than 25% sequence identity. Yeah?
There is no twilight.
The problem is I cannot study the intrusion into the twilight zone if I don't have pairs.
Actually, I would expect something like 70%. When you're saying that.
Redundancy reduction you can do in a different way. Yes, that is fair enough. That's one way in which we could do it. Another way in which we could do it is essentially you take out the redundancy reduced and you line it against everything. So somehow you keep, and this everything could still be 100% reduced or 80% reduced or something like that. Yes. Okay, so the story is set up. What I'm going to show you now is number of residues aligned. So how long is the alignment? Percentage sequence identity up here. Red are proteins that have different 3D structures. Blue, proteins that have similar 3D structures. In gray is a fit, if you want. And the fit is such that you see the gray line is such that most of the red points, or most of the red points, this is what we have to get back to the notion here. But you see that somehow visually red is below the line. Blue is substantially above the line, right? So above the line you find, it seems, a lot of blue and not a lot of red. Okay? There is some issue here. The issue is we see visually a lot of points, and what I didn't say, it's somehow implicit in what I told you so far. This is a very heavy data plot. So this is done a while ago. It was not as big as it would have been today, but this is 10 million points. Okay? And you clearly cannot count the 10 million points. You can't see the 10 million points. And that leads to the point, so this looks like one of these 10 million here. But the problem is that from what you see here, you cannot gauge how big is the red above the curve, right? What are these numbers? We have to, so this is one way, you have to visualize your data in as raw a way as possible, but that's not the only way you report. You have to really count. Okay? And one way of counting is we take this curve here and now compute a distance from this curve. There's a distance in blue here above the curve, and red is the distance below the curve. So you plus 5 is, so you shift the line by, the line now is blue here, you shift the line by 5, 10, and so forth, right? So the further away from the curve, the closer you essentially are to 100% sequence identity. Okay, and now I'm going to show you what these numbers look like in raw numbers intruding into the twilight zone. So in terms of sequence identity is what I showed you before, 35 to 10%. But in terms of this curve shift here, distance from this curve is plus 10 to minus 15. 0 is the curve. That is a logarithmic scale. That is the number of pairs of proteins that are similar in 3D structure. Okay? Can you please observe? Can 2 or 3 people say something that they see? So one thing that you have to do, one way of approaching your data when you write it down in your thesis, is you look at the graph and you describe what you see. That's typically in the paper the results section. Can I have a few descriptions that you would put in a graph like that from the room? Everybody, just one observation. Or yes?
The number of protein pairs is decreasing, the distance to the HSSP threshold increases.
So the number here goes up. Yes. Next observation. Next person. It's something you have to train. You're not born with it. Just yeah.
The sequence identity strangely is like inverse with another protein pair.
Oh, but so again, when we go down here, we go to more distance. No. So this essentially, in terms of sequence identity, you going down means more distant relatives. So you go more from daylight zone into twilight zone into midnight zone. You go towards 0. So again, let me put at 100 aligned residues, the curve is at 33%. So if you had 100, let's look at the saturation point here. It's 400, it's at 20. So the 0 point here is 20% sequence identity at 400, or I don't know what so this axis I looked up from a different, is it, yeah, that axis, no, I don't know what, it's somewhere 150 or something. This is an analogy that holds for a particular length. It's a cut, so this axis relates to that at a particular cut point. Anyway, going down here in this direction, or in terms of sequence identity in that direction, is the same thing. Going in this direction here means to the left means the pair of protein is more distant, distantly related. And again, the observation previously was that the further they diverge, the more pairs you find. Okay, here's another important observation, and this is exactly what you have to write in results. It may be absolutely trivial for you, but it's an important one. You go through an order of magnitude here. So the statement before was true. Blue increases, but not only does it increase, it actually jumps by more than an order of magnitude. So this is a logarithmic scale. You go from something like 7 times by 10 of 3, so 7,000, to something that's in the ballpark of 10 million, of 100 million. So it's a huge increase, right? Over the span that I see here. It's not only blue goes up, it goes up radically, yes? Can you try?
I don't see how the curve is going up.
So the curve here is lower than the curve here. That's the way now, is that? Going this way, the point here is higher than the point here. The curve rises. You may argue that here it no longer here, at some point it stays constant. That is true. But essentially the curve rises all the way at least until here. Right? And again, since this is a logarithmic plot, this is actually more than an order of magnitude. It's not, so I'm a little bit at a loss what to tell you. The point here is lower than the point here. Yes?
Yeah.
Then the curve goes up.
I can see that, but I think I just don't understand the graph very well.
Okay. So this axis here shows the pair-wise sequence identity. So I compare two sequences, two protein sequences, and I ask what fraction of residues are identical between these two pairs. That is essentially plotted on this axis here. Now here I also put another feature, which is the length. And why do I put that? Because when I have two pairs of protein that are 100% identical, and that 100% identity is over 10 residues here, that does not tell me anything about structure. This is just randomly possible, so two stretches of 10 that are identical, two words of 10 alone are not enough. Okay? That's why the percentage sequence identity alone is not enough. I have to relate it to the length. I should have said that more clearly, I'm sorry. And since I sort of need both number of residues aligned and percentage pairwise sequence identity, I somehow now have to construct a new function that considers these two variables and sort of merges these two into a function that spits out one value, and then I can look at distance, this one value, and on the other I look at accuracy, or pairs of proteins that are similar or whatever. And the way I do that, I take this curve that somehow did fit the original data in some sense, okay? And I'm going to look at the distance from this curve. So I look at distance minus 5, minus 10, minus 15, minus 20, plus 5, plus 10, plus 15, plus 20. So plus 0 means this curve, plus means more similar than the separation line, okay? So that means that this here, plus 5, plus 10, 0 is the curve, minus 5, 10, 5 below the curve, 10 percentage points below the curve, 15 percentage points below the curve. So it means that I go from right to left. For the pairs I find on the left, they have much less sequence identity than the pairs on the right. Up here, I just, for orientation, put now the number of percentage sequence identity that somehow it corresponds to. Okay, so we go from 35 to 10, and again, where exactly this number comes from depends on where I cut in this line here. But again, the x-axis means I reduce from right to left, I reduce sequence identity. Now the y-axis. On the y-axis we simply have pairs, number of pairs. Again, I have a 3D comparison and I have a 1D comparison. 1D is sequence, 3D is shapes. And I'm trying to relate to get the function that relates one to the other. And I begin, on the first step, I just observe numbers. And what I observe is going from high levels of sequence similarity to lower levels, the pairs of proteins that have similar 3D structures increase. Another way of sort of saying that is there's a lot of blue below the curve. So 0 is all the blue lines up here. Oops, I should have put these closer to, that is 0, okay? That's all the blue. This number here, I don't know, 2, what does it look like? 2.5 by the 25,000, roughly. So what you saw in the other plot above the line was 25,000 blue pairs. That's what this one says. Is this one clear to everybody? Okay. Now what we need is of course the red ones. So pairs of proteins that do not have similar structure. And again, you clearly observe that red goes up. But not only that, red goes up by, so I said blue increases by more than an order of magnitude. Red goes up in this region here by 5 orders of magnitude. Right? Another way of putting that, so at the line of 0 here, we have already a lot of red. So 0, in fact, the HVal of 0 that I showed you there is not the best distinction. There is a lot of red visually above the line, right? That's one thing you see. You also see that if you are at 10, at a distance of 10 here, 10 would be this point, this line here. At 10, you have 0 red. Okay? At some point of roughly 3 here, you have 50% red and blue cross. So roughly somewhere here at this line here, essentially all you find is 50% right. Okay? So what this one shows you is that you want to intrude into the twilight zone. So again, let me repeat. Daylight zone is defined as red is 0. So the daylight zone is right of this plot, or begins possibly exactly here, where we have 10 in this plot. So what we see here is we do want to enter into the twilight zone because the blue increases by more than an order of magnitude. If my sequence alignment is done in order to find pairs of proteins that have similar structure, I have to get into this realm. I cannot just stop here, because what I find before that point is just not enough, or not many. Many things of the ones that I want to find. I want to find as many of the 3D relations as possible. I can find, yes?
This y-axis, number
yes.
of protein pairs, this is meaning having similar function, right?
This means, well again, what I plot here is the pairs of proteins that have similar structure. Structure is a proxy for function, but in the course of this first lecture, mostly we consider structure.
You should rephrase it as 3D structure.
Yes, 3D structure. So let me go back to the way we defined that. So we compared the 3D shapes, and we said that if they are similar below a certain cutoff distance, we call them the same 3D structure, and if they differ very much, then we call them different structures. In between the two, we ignore them. So this again is red, and this is blue in the following. Is that a question? Can you ask it?
I'm not sure what red and blue is, actually.
Yeah. Thank you for asking. So, I believe I'm missing a slide here. The PDB, Protein Data Bank, is a repository that contains all the proteins for which we know the experimental 3D structure, the shape. So we have about 100,000 shapes. Now what we do is we do 100,000 against 100,000, or some subset comparison, we will later say, right? See. So we really look at the three-dimensional shape, and we ask the other 99,000, again we're going to reduce, redundancy reduce, so 10,000 against 100,000. So we take the first one out, ask how often do I find, what are the pairs where the shape is the same? And if they are really, really similar in shape, I say they have a similar 3D structure. In the following curves, this means the blue. Okay? When they are very different in their three-dimensional shapes, I say they have a different shape. In the following plots, they are red. In between, I throw all the pairs out because I'm not sure. Are they similar in structure? They have some similarities, some differences. I'm not so sure, I throw them out. But I only take the very clear cases. I label them in the subsequent plots here. They are labeled blue and red. Blue and red. Red, different structures. Blue, same structure. In terms of counts, red, different structure. Blue, same structure. Again, I want to infer that two proteins have a similar structure, and I want to find proteins of similar structure, as many as I can, from sequence. If I stopped here, where there is no red, I would find roughly 8,000. If I could go a little bit further, I would find 100,000. So stopping here where it's safe in the daylight zone, I miss more than 92%. Okay? And the real story underneath this is even more dramatic than what I show here. This is the incentive for trying to go into the twilight zone. Now, so, yes?
How does it look even further to the left? Because it can't go on forever.
No, so at some point it will be zero, right?
Yeah.
But you begin to see that there is sort of a saturation point here. It grows a little bit, but again, so most pairs of similar structure are actually sitting somewhere here to the left. It hardly grows, it goes a little bit further up. Not much, yes?
There are so many pairs of similar 3D structure having only 10% sequence identity.
Yes.
Does it not somehow hint to the fact that something else might drive 3D structure, or this is against the old rule?
Okay, so how come? So we obviously go to a point where we have a lot of pairs of proteins that have a similar 3D structure without any sequence similarity, or essentially very little. In fact, I am skipping at the end of the lecture today the real examination of that is the midnight zone, and the midnight zone is highly populated. So how come? Is that not sort of beating the idea that sequence codes for structure? I cannot prove to you that the answer to your question is no. Because we cannot predict 3D structure from sequence. But we cannot identify the sequence or the reason why these two structures are the same. But we have seen examples where we can predict the structure from sequence for two proteins, and they look similar, and we cannot see the sequence similarity. So that would suggest that I can sort of pick up the theme, but I cannot pick it up in a sort of understanding way. I cannot write down the equation. The molecular dynamics simulation can give that in some cases, which suggests that there is a theme, but we are not able to identify the theme. Okay?
I'm a little confused whether we're more interested in the sequence identity or the 3D structure similarity.
So the confusion here is, what is it that we want? Do we want sequence identity, or do we want a structure similarity? Ultimately, we want to understand structural identity or similarity. We want to see whether two proteins have a similar structure. But we want to know that for proteins for which I do not know the structure. Because the structure costs $100,000, and I cannot do it. The sequence comparison, yes, sometimes I said dynamic programming is slow, but you know, for any pair of proteins that is really, really fast, and I can do it. Sequence comparison is very, very simple. So I want to relate something that is simple to something that is more complicated to do. Okay? If what I here for the blue and the red, I have all the structures, I can do all of that. All of that is simple. But for most proteins, we do not know the three-dimensional structure. We could not do this. For most proteins, however, we can compare with their sequences. So sequence identity is what we can do for any pair of protein for which we have a sequence. Sequences are cheap. So it costs about 50 cents per protein to sequence it. It costs about $100,000 to do the 3D structure. So sequences are sheep, and that's why I want to find a relation that brings me from something that I can easily do, a sheep comparison of sequences, to something that I want to infer similarity in 3D structure without knowing the 3D structure. Okay? And that's why I'm sort of building up the curves, the functions that bring me from one to the other. Is that clear?
Could we go quickly back to the chart where revenue was separated? So basically, each point on one of these charts is an alignment between two proteins, and this gray line is just like a needle fit.
Yes. So again, I repeat the question. So every point in here is a comparison between two proteins. Yes. The coloring means I compared their three-dimensional structures and I found if it's red, they differed. If it's blue, they were similar. Okay, the gray curve now was the question, is just a mean fit. Well, yes, that is one way of saying it. The gray curve did not evolve as a function that you can actually write down. So there is no software that you can say fit this and give me the gray function. The gray function evolved by some intuition and some math of sort of computing the background problem. So what's the random probability of matching two sequences? And that sort of gets a certain exponential function, and this is sort of a play on the theme of trying this particular type of exponential function to fit to the data. So it's a mix between empirical computation of what should a change be like. But it's a fit, yeah?
If this holy grail is found, if somebody tells that someday, okay, I can now 100% infer the 3D structure similarity from sequence, will the procedure be like, okay, we have like 100,000 proteins almost for which we know the structure, so we compare a new protein to all of them and infer its 3D structure?
Okay, so this question is very easy for you, very important for you to understand, because it skips an entire lecture. So the question is, let me get to that point. Well, if we found this relation that relates sequence identity or sequence relation to structures relation, then could we not take the 100,000 known structures, apply this threshold of, I'm just finishing your question because I suspect where it's going. Apply this sequence function and find all the proteins that are similar in terms of structure to the ones that I know the experimental structure of. Is that your question?
Actually, the end of my question was, if this is the case, wouldn't this be terribly biased towards the 100,000 proteins that you know? Because they are not...
would be completely biased toward the 100,000 proteins. But could I do that? So I thought that was your question. So could I do that?
If you want to compare another 100,000 proteins that you are very interested, so 100,000 by 100,000 doesn't sound like a big competition.
Oh, no, wait. So if I found this, we call it holy grail, that relates sequence to structure, I start with 100,000 and I find everything in the database of 85 million that is similar to these 100,000, right? And that could be a number much, much larger than 100,000. And if I found those, then I would find all the proteins in the 85 million for which I somehow sort of can guess the 3D structure. And that does work. And that is called comparative modeling, and we're going to talk about that next Tuesday. In fact, that's the only way in which we can safely predict structure, and that's the most successful way in which we can do it, because we can actually apply this for over 30 million. This gives us a prediction for 30 million. Out of the 100,000, we get 30 million predicted structures. And it's as simple as that. Now, here, the real question was, isn't that biased towards the 100,000 we have? Yes, absolutely. How much will that change with the next 20,000? And that is a complicated question. So the argument is out there that not much has changed in the structure over the last 10 years. And that is by sort of chopping it up and finding sort of substantial motifs and saying there is no new motif discovery. Maybe true, but we still cannot take a particular protein and predict which of these shapes will it look like, or which of these shapes will it connect. So in that sense, so this is a debate that I want to avoid. We can discuss offline. is more complicated. So yes, there is a bias. How much that really matters is unclear. Maybe I have time to talk about the dark proteome. So the dark proteome is work where we are essentially trying to predict the things for which we do not know the structure. Do they completely look different? Is there something out there that we haven't seen so far? We call that the dark proteome, that would look completely different. And we believe yes. So we have evidence for that in machine learning. Let's just leave it there. Any other question to this part? So I believe what I left you with is this idea that we have some idea about this relation from sequence to structure, that it's important, and that we want to intrude into the twilight zone as much as we can. And intruding into the twilight zone as much as we can brings us to the next issue. So far, we have always discussed pairwise alignments. We took two proteins, compared their sequences, and asked whether they are similar, whether we can infer structural similarity, for instance, right? Multiple alignment is the idea of, instead of taking 2, take a higher number. For instance, 3. 3, you can still sort of do dynamic programming, so you can do a brute force. In fact, I haven't seen the paper myself, but the rumor has it that we can really do 6, so we can align 6 proteins. So I put a cube there, a 6-dimensional object it would have to be. Which is difficult for me to show. But it takes a 60 terabyte mainframe machine and it's unclear whether it really works. The problem is, today we handle families that have thousands of members. So whether we can do 6 or not doesn't really matter. We need a solution that goes much larger than 6. How can we do that? And ultimately, we all live off a hack here, and the first hack was introduced by essentially Rost Doolittle, and the way it works is the following. I take, in this particular case, 4 proteins. I do all the pairwise comparisons, do an all against all, and the numbers here are the percentage sequence identity for the pair. So A to B is 90, A to C is 80, and A to D is 70. That gives me a sorting. So now I begin to take the first pair. The first pair is the one that has the highest sequence identity. So I could do AB or I could do BC, but let's start with AB. So those are the sequences underneath. And the way I'm going to do that, I'm going to simply align A and B, the most similar pair. Okay, in the second step now, I do the other pair. So we have only 4 sequences here. There's the second pair that I do. And that essentially gives me 2 alignments. In these 2 alignments, somehow there is something that they have in common. Here, sort of consensus sequence, and there's one here. And now, in step 3, I align these consensus sequences. That's one hack. Is it clear? So essentially, I boiled down the problem from comparing 4 proteins into 3 pair comparisons. Pair comparison I can handle. Very, very easy. Now, I could have done it slightly differently. So I could have done the first step, but in the second step, I could have, instead of taking the next pair, take the next sequence here, the green one, and align it into the first pair. So the first pair here gives a consensus, and I simply take the sequence 3, the green one, and align it into that consensus. So I have different ways of doing that, yes?
What's the rule for creating this consensus?
So again, this is a very good question. So what's the rule for creating this consensus? There are many ways in which I can do that. I can say, well, it is trivial for the residues or for the amino acids where they're identical. There the consensus is immediately clear. Where they're not identical, I have obviously a couple of choices. I can say I take the first, I take the second, or I do X or one or the other, but that is more complicated to handle, so I have to make a decision. I have to really put a letter. I can put the letter that is the most informative. So which essentially could be the least often occurring residue, or there are different ways in which you can do that. In the one I chose my example such that I put the first. But again, so there are different ways of handling it. Yeah?
Just quickly repeat what stuff we use.
So step 3 in this particular case, again, there are different ways in which I can do that. In this particular case, step 3 is to take the consensus sequence that I have here and align it against the green. Oh, step 3 is take, step 2 is the green here, that's this one. I take the consensus sequence from step 1, align it against the green, get a new consensus sequence, take that new consensus sequence and align it against the black one, 4, the protein D. Okay? And this is again, so the reason, if I had either this letter or the other letter for the consensus sequence, then I would not easily know how to handle the alignment of the consensus sequence. In principle, I could do it as a mathematical, but in the simplest one, originally it was done like this. Again, it's a very simple hack. The downside is a little bit, we have a lot of possibilities to do it. And it's not really clear which one is best. Up front, they look equally good or bad or whatever to me. And this brings us to a sort of different concept, the concept of a profile. Now let's get into the story that I showed you before, where you build up sequences. So you take the 2, you align them, and from the alignment of the 2, the first 2, you bring a new one in. You begin to see that there are some regions that are more conserved than others. And by just looking at the third, you see that there's something that here is like an insertion, that the Q here is important. So bringing in more residues, you begin to evolve what I showed you before. The situation where you see some proteins are sort of long, other proteins are aligned only to this part, other proteins are aligned to this part. In the first lectures I talked to you, showed you these kind of plots, and I essentially called them, this is evidence of domain. But it's also evidence that when you look at more than pairwise comparisons, a certain structure evolves. Structure, I don't mean 3D protein structure here. A certain image evolves, that you have to keep some positions more importantly than others. And that ultimately, in this slide, and the following slides come from Teresa Worth. So ultimately, this is a theme that has to do with propensities, with motifs. So here is a motif for a transcription factor binding site. That's position 0. This is left of the binding site, right of the binding site. And this is nucleotide binding. So you see 4 letters here, the 4 nucleotide sequences. The height of the letter is proportional to what you observe. So what you immediately see is a motif here. You see that at that position -6, you will always have a T. At -7, you will almost always have a C. At +6, you will have a C, and so forth and so on. And you can easily write this, what you see here, into a regular expression. That's one way of expressing it. Or you could write it into, we could write it into a substitution matrix. A substitution matrix essentially says which amino acid do I put at which position, but now the substitution matrix is one that is specific to a particular place at that position. So I'm no longer saying, well, doing a D on an N gives me this much point, but doing a D on an N on a particular place. Okay, this is the idea behind this. So in order to get to that, you begin with the sequences, you convert them into a frequency, so you count at every single position what do you observe, you do a relative frequency, you get that into an count matrix, you get it into an entropy. In the entropy conversion you need something that is called So you need some spurious counts here. You need to add some values so that you don't get infinity here. And then ultimately you get what is called a profile. The sequence runs from the top to the bottom here, from 1 to 49 residues in this particular case. This would be, for instance, the consensus sequence. So this is alignment, or there are 4 sequences aligned here. And at every single position, so the first position, there's an E in protein 1, a G in protein 2, a V in protein 3, and an L in protein 4. and so forth. We go forward. So this implies an alignment. This alignment implies a substitution matrix that is specific to the place. So you get a certain probability here to have an alanine at the first point. You get certain, by the way, so consensus sequence here, you have an example where the V is the most informative one. It's not the first one here. Consensus sequence here is written as V. Here's L because that's the dominant, most informative part. Anyway, so at every single position, you have a certain probability to put either of those 20 residues. And the probability changes with adding more sequences. So the more you build up your multiple alignment, the more the positions evolve to have a certain profile. Okay? The idea is clear, is it? So we go from where we ended in the last lecture, which was a substitution matrix that is for any pair of proteins, without knowing anything, to one that is specific to this protein. So in order to do this alignment here on the left, I take the blossom matrix, but then I build up the alignment, and by building up the alignment, the position at every single point here, position 11, it is MLII, and that gives me a probability. Now comes protein 5, and protein 5 has a high propensity to put an ML or II, and the highest for I at that point. Okay? So this is no longer, I come with a new sequence, protein 5. I will no longer use blossom, I will use this specific matrix. Okay, there were two questions in the room. Yes?
Can you explain again how to read this table?
Yes, I'm sorry. Protein, that's the number of the residue. The first residue, second, and so forth to 49. This is the first protein. The sequence of the first protein. The sequence of the second protein, third, fourth. Using pairwise alignments, so one way we start with this one and then we line this one into it. Then we start with this one, you line this one into it. Start with this one and you line the third one into it. Builds up the alignment that you see here. And then you simply take this alignment. The alignment implies here that at every single position, for instance at position 11, the first one has an M, the second one an L, the third and the fourth have both I's there. And that you transfer into probability matrix. Probability matrix reflects what's the probability of at that particular position having an A, B, A, C or something else. Okay? Is that clear? There was another question? Let's run away, yes?
So the order is, first you have like 4 independent proteins from one family, then you make pairwise alignment and you get this probe, then you build the matrix, and then you come up with the consensus, right?
That is correct, yes. And hearing your question, I realized that that this slide is very complicated. Okay, let me repeat the question just to explain. There's a lot put into this. I start with my first sequence here, one protein. And I align it to another protein. For the alignment I use a blossom matrix. That is the second protein here. Then I do the same thing, I align it to a third protein. And I align it to a fourth protein. Then I superpose these 4 alignments in the way that I showed you before, these hacks. And that gives me the first 4 columns here, which I call an alignment of 4 proteins. Alignment means essentially I know what position 1 in the first corresponds to position in the next protein. Or there is the G at position 17 in the first protein, that corresponds to a G, S, and Q in the other 3. Once I have this alignment, I can transfer what I see, the observation, into counts. I observe 2 Gs, 1 S, and 1 Q at that point that I just had, and can transfer these counts into a probability. Log odds ratio, and that's what you see, the position-specific scoring matrix or profile. Once you have the profile, you can actually write down the consensus sequence. Because in the consensus sequence is the most informative letter. You need the profile for that, yes?
If it's not too specific, could you please again explain in a little bit of detail how do we go from like observing 4 characters here to this matrix by means of this colorful, table of letters. You have this 20 positions, colored, multiple size colored letters and previous ones. And this is the essential to view the matrix, right? Can you repeat? Are we using this?
Now I know. Yeah. You talk about the transcription factor binding. This. Oh, wait a minute.
Yes.
It's the animation of the slide. Yes. So how do I go from this one to the one before? So this one here, ultimately, remember when I put the 4 proteins, at every single position I can count how many I have of M's of whatever letter, right? And this is exactly put here. The height of the letter is proportional to the count. And in the slide behind, I have the count. Now I take the count and I transfer the count into a log-odds ratio. And that has to do with the background probability. Okay? So ultimately there's the 1-letter code, there's any amino acid disallowed, blah, blah, blah. This is just a regular expression. And I can take the regular expression and turn it into account by beginning with a regular expression. With the observation, looking at the frequency, the absolute frequency, getting a relative frequency, transferring that into probability, and going from the probability to log-odds ratio, where I need pseudo-counts. Here is still not pseudo-counts in there, that's why there's infinity. Pseudo-counts is I'm going to invent some counts, some low counts somewhere, so that I don't get infinity in the log-odds. And that ultimately is what you see on the right-hand side.
So the consensus is the most frequent letter from the code, right? So for example, in position 3 you have all B's, and therefore the consensus is B. Yes,
the consensus is the most frequent typically, but it's actually also, here you see an example where when no pair of protein has the same letter, you use the most informative letter, the V. Most likely the V, maybe I made a mistake, but most likely the 6 here is the highest number in this.
It is correct, but then for number 7, it's D. Why is that?
It's D, and again, yes, because we have to get back into the, where did I give you the last slide? Nope. Here. Dimensions. D. No, that is not explaining it. So there is, I'm afraid that the answer can only be that this slide is not giving you the complete information, which I defend by the fact it's not my slide. But 7 is a very good observation. Most likely this is not compiled on exactly those 4. Most likely this is compiled on a larger set. Sorry for that. So I have to take that out.
But yeah, as the idea is the most frequent letter is what gives the most information. That's why we use that.
Yes. So the 7D is absolutely not explained here. Yes.
You could have used E, which is 7 as well, but is in the sequence, right? So if you check the scores, the score for E is 7 as well.
Yeah.
And it is in the sequence.
Yes. So I have to correct that. I would be a hacker and use the same slide. Yes. Thank you. Yes.
So how would I select those proteins I used to build this specific matrix in the first place? And how would I decide later on to use this specifically?
So the question is, how do I find which proteins I put into my alignment? How do I define, or how do I decide now I stop? And we are back to this issue here. We essentially will include anything for which at some level we can, so there are different ways of playing this, of doing this. One way of doing this is I define a threshold at which I am sure to infer the 2 proteins have similar 3D structure. So I stay somewhere right of this line here. In this particular plot that would suggest a value of 9. I should pick as a threshold the 9. As long as I have a 9, I don't make a mistake. So I take everything that I find until a threshold of 9. And those in the other slide there would be the 4. Everything I find, so I take my sequence, align it against the database, I find 4 proteins above 9. So then I decide I include those 4 proteins. Those would be, hypothetically, those 4 above 9. Answered? Anything else? Sorry, yes?
Can we go back to the slide of the A, B, C, and D proteins?
ABC, which one?
The one of the A, B, C, and D proteins.
Is that the one?
So in step 1, we were computing the consensus of proteins A and B. Right?
Yes.
So there are 2 amino acids E and D, and we decided to choose E in the consensus.
Yes.
So why did we choose E and not D?
Again, so I presented this sort of multiple alignment, build up a pairwise alignment as a hack. And I told you that there are various points of this, doing the hack, there are decisions that I have to choose. And they're arbitrary. So one decision could be, the simplest decision here would be to simply take the letter of the first. Right? Or, so let's get back, there was a question before. So all the possible ways in which I can do that is I can take the letter which letter will be here when they differ. It can be the first, it can be the second. Since we cannot align things that are regular expression, at least in the formalism I've introduced so far, we cannot easily do that. We need to choose one letter. So it can be the first, it can be the second, or it could be another letter or another choice that is done by some other criterion. The other criterion that I presented was the most informative of the 2. Or in the over next slide there, the famous D at position 7, it could be in some matrix the most informative letter. And it may not even be any of the ones there. Would make a lot of sense though. So there's a limited number of decisions, limited number of choices I have. First, second, most informative of either of the 2. Right? Is that clear?
Yeah, but I don't understand the point.
Why you do this? We can align pairs. I give you a string of letters. I give you 2 strings of letters and I know how to align them. Okay? And I want to align 5, or in this particular case 4 proteins, right? I have to find a way to map the problem of aligning 4 into pairs of solutions. Because I have a solution for 2. So how can I take the 4 and do it in a way that I know how to do, meaning pairs? And building a consensus sequence is one way of doing that. Tell me another way of doing it. Anybody has another way of doing it? So again, I want to align 4 proteins and I want to map it to pairwise alignment pairwise alignments because that's the only thing I can do. Do you know a solution without using condensers? Sequences. Yeah?
So you take the protein C and D, find the similarity, and then compare that similarity with the protein C, and then you find their similarity and then the D. That's what you do.
Yes, but again, so what you now, this is what I did in this step. In the one before, I showed you a different way of doing it. Yes, whether you do the first 2 and the second 2 and do consensus and then do consensus of consensus, or you do the first 2, do consensus, take the consensus and do the third in there. Those are alternatives. It's the same idea, it's just a slightly different way of doing it. Right? But there is no other way of doing it. You can only do it with the consensus. The only way of doing it differently is you have to introduce regular expressions, but then you have to find a rule how to compare irregular expressions, or you have to translate regular expressions to probabilities. And then you have a new mechanism, and we will possibly get into the new mechanism. I believe I was a little bit over-optimistic about how much I can do today. So some of you may observe. Ultimately, let's just fast forward to that point. Hidden Markov model is sort of a generic way of doing it that does something like that without consensus. But then you really have to come up with a probability theory. And I will present, I will roughly get there. At least to the idea. So PsyBlast is the blast is the fast hacking. So again, here we have such a profile at one particular position. There's always, so the purple shows 4 very conserved positions, and ultimately this is the generic, the Blossom matrix for aligning 2 proteins. You take Blossom. Once you have more, you take this profile, and the concept of the PsyBlast is you begin with the fast hashing, the words I showed you. In between the words, you extend by dynamic programming, and again, this is so fast because we speed up, because we compile it, we cut the length. Then we do the statistics, compare all the pairs, and build a profile. Just the same way that I showed you in the previous slides. In the previous slide, we had 4 pairs, and I build up a profile. That's the way we do it here. Once we have the profile, we now sort of illustrated here, we take a sequence and align it against the profile. That means for the blast, what we do for the PsyBlast, we iterate over it. So we build this profile by searching everything that maps above the threshold of 9 in the database, build a profile of that, and then we do another step of doing everything again in the database, because this time we may, through the profile, get deeper. We get below 9, because now we know, in the profile we know that particular positions have to be conserved. And this is no longer the sum is 9%, but this is, at that position you have to be conserved, all right? And then you can let go for other things. This is ultimately the idea of PsyBlast. So there is a sequence-to-sequence profile. The result of that, sequence-to-sequence comparison, pairwise sequence comparison, the result of that is a profile. And then there's a sequence-to-profile, or profile-to-sequence comparison. And the result of that is an updated profile through iteration. That's the idea. Then there's a method called ClustalW that sort of does the same thing that I showed you before. So it does a tree mapping, and it does it in a very good way in dynamic programming. It uses a phylogenetic tree, gets many of the details right. Then there's the hidden Markov model, and that, again, these methods are trying to get deeper into the twilight zone. Hidden Markov model, essentially you have a state, 1, 2, and then you have a transition probability to another state, transition back to itself. For instance, here is the example for football, and it's from the season a few years ago, a particular team in the vicinity of Munich playing Augsburg, and it shows the score, it shows the home team, when they play away, it shows the number of people in the stadium and a date. So ultimately, the hidden Markov model here would have a score, 3 states, the win, draw, or lose, and we would try to predict this state given something else. They sum up to 1, and what this something else could be, it could be that we actually look at whether it's a home or away play. So in the simplest model that we could possibly say, we could say when they play home, they win more often than when they play away, right? So that would be a hidden Markov model that simply as input has home or away, and then comes up with a probability that then, if they're home, they win. Okay? And it compiles from the data all the transition probabilities. For doing an alignment with this, you essentially, again, you have the letters, and then you get into various states. Here's another example for this. The traffic light, where we have red, yellow and green. This, however, is deterministic. This is not a hidden Markov model kind of situation, as opposed to the weather, where you go from sunny to cloudy to rainy, and that is a probabilistic transition. And this is the kind of thing that you exactly do by the hidden Markov model, and you try to do it in the first order hidden Markov model. That means that the next decision is based only on what you know today, what you know now. Using it for alignment, you have a start position, then you get into an insertion, a deletion, or you get into a match state, and so forth and so on. And then all of these transitions here, you learn from a set of proteins, again chosen by above a threshold of 9. You take everything above a threshold of 9, you throw that into a pot, and that in fact optimizes this model. Okay? In the optimization of this model, you begin at some point. Before any amino acid is aligned, we expect that we have essentially the background probability. Then we get to a particular position, then we get a new entropy, we read off. So this is our first step alignment. That first step alignment gives new probabilities for every single position, builds up this matrix. It's the same thing that I showed you before, except for now it's put into the hidden Markov model. And voila, we have 112 slides, so we fast forward quickly. There's another idea in which we can do it. We can do the genetic algorithm. You may have heard about that. So one issue with the genetic algorithm, or an important issue, is the independence assumption that we had so far. For whatever I showed you so far, the dynamic programming always works if and only if what happens at position I is independent of what happens at position I plus 5 or j, where j is not equal I. So the alignment, what influences the alignment is only the information of what happens at position I. In real life, in real proteins, that is not true. So 99% of what people do in their alignments assumes that I and j are independent, and they are not, and it works very well. The only method that really allows us to drop this assumption is the genetic algorithm, because it operates on segments. So here's one example for genetic algorithm that in fact optimizes the shape here of this antenna. And the way you do it, you have a bunch of events. So in this particular case here, these are the two parents, the child, and you take some from here and some from there. Okay? And then you mutate. Those are the simplest rules in which you can sort of do the genetic algorithm. For doing it for proteins, you begin with a library of local and global alignments. So you have a bunch of methods that you apply. You use a hidden Markov model, you use dynamic programming, you use PsyBlast, and you put all of this into essentially local alignments. And then you put these local alignments into the genetic algorithm. You do a child, you create children, you create mutations, and you create, through this genetic algorithm, a new alignment. And that method is called T-copy. Again, it uses multiple alignments, it uses local alignments, global alignments, it can use structural information, it can use specialist information. All of this can come as simply to phrase constraints that have the fitness function of the genetic algorithm, and then you crank the handle and you get out the best alignment that uses all of these constraints. Now, the sequence, so only using sequence information, pairwise alignment can intrude into the daylight zone. Sequence profile can go into the twilight zone. And profile is sort of the next step that can go zone further. Profile now is the idea. So you have, for one protein you have a profile like that, for another protein you have a profile like that, and now you're comparing the two profiles. Again, in terms of probability, it's very straightforward. So asking whether that particular column fits into that particular column, you simply do the cross of the two vector products or something like that, right? Again, the problem here is that you could do many choices for or something like that. It's a simple vector product. You can compile the vector product in different ways, and that gives you a lot of possibilities, and that is in fact the problem of profile comparisons. There are too many free parameters. Nevertheless, there is one method here that comes from from Munich, H.H. Blitz, from the LMU, that essentially uses exactly such a profile-profile comparison through hidden Markov models. And essentially we're back to the idea of consensus sequences in some sense. So they use simplified, a slightly different alphabet of the amino acids and put it into something that is like a consensus sequence to gain speed. And with that, we sort of get almost into the midnight zone. And with that,